Hi, welcome to this production of the Institute for the Meanings of Science. My name is Samuel Longcar. I'm the Institute Director and founder and the editor of the Marginalia Review of Books. I'm privileged to be able to present the research director of our Meanings of Life, the New Biology Project, Philip Ball. Dr. Ball is the author of How Life Works, a user's guide to the new biology, and a longtime editor of Nature, and the author of over 25 other award-winning books on science and history. We're very privileged to have today as a special guest for our second conversation, hosted in partnership with the Francis Crick Institute, Dr. Naomi Moris. Dr. Moris is one of the world's leading developmental biologists. She received her PhD at Cambridge University and is now the group leader of the Moris Lab for developmental models at the Francis Crick Institute. We're delighted to host this conversation. Thank you so much, Dr. Moris, and thank you, Phil, for hosting us again.
Thank you, Samuel. Okay, well, Naomi, I wonder if we might start with, actually, maybe I should sort of hark back to the conversation I had here with Paul Nurse, because we are interested in exploring the narratives that we tell about this question, how life works. And with Paul, I discussed molecules and genes, which is sort of what he does, and I feel like what we will be doing, what I imagine we'll be doing, is zooming out to another level and to look at really the morphology of the whole organism, how the whole organism takes shape, which is really what you look at. And in a sense, that's the older question, because before we knew about genes and molecules, we knew that there were embryos, we knew that they took on a shape somehow. That was a subject that even Aristotle was interested in. And there have been many different ideas about how that happens. It looks kind of magical, of course. We sort of see this thing taking shape from something kind of unformed into something that has the shape of the whole organism, whatever that is. It seemed that in the 1950s, when molecular biology really started to take off and when our understanding of genetics started to take off, that we had an answer to that question finally, that it was somehow the expression of a blueprint in the genome. Now, obviously, the genome has to be pretty central to what's going on, but the story that's developed since then has, I guess, proved to be rather more complicated. And, whether we can still cleave to that basic picture that all the information is in for the whole embryo, for the organism, is in the genome, or whether we need a more nuanced view of it, is really what I want to explore. And I wondered how you would talk about that question. If there is a plan for the organism, where does it come from? Where is the information? Or is it the right way to think about it at all? To think that there is some pre-formed plan that is simply, executed in some way?
Yeah, I mean, it's such an interesting question, isn't it? I think it's become clear that there is no kind of higher plan in that sense of something that is instructing the formation of the body plan. I mean, certainly, if we looked at the mouse genome and the human genome, it's not clear to me why the human genome makes a human and why the mouse genome makes a mouse, right? I don't think that we could predict from that sequence of letters the body shape of a mouse or a human or any other animal for that matter. So, yes, in some sense, the genes contain information, but they're certainly not this kind of blueprint architecture that is somehow telling the embryo how to form the structure. I guess I would think about it more in terms of rules, perhaps, or logic that is encoded in some way. Often, obviously, we think about genes, we think about genetic networks of genes, so not just the actual genes themselves, but how they relate to one another. And somehow the cellular interactions themselves have to be following certain rules. And by doing that, then they end up with complexity and beauty and intricacy, and all of that comes from the rules rather than the actual blueprint. But it's definitely something we're still trying to unpick, and I don't think we're there yet, and certainly couldn't tell you exactly how everything is formed.
Well, I guess one of the aspects of that process is that it's not simply about this massive cells finding a shape of some sort. The cells themselves, I mean, it starts off with a single cell. As they divide and multiply, they also have to become specific tissue types.
Yeah.
So cells have to, if you like, to decide what kind of cell they're going to become. And that seems to be a crucial part of the process. Can you walk us a little bit through that process? How does a cell in a very early embryo decide, I'm going to start on the path towards becoming a neuron or a skin cell?
Yeah, I mean, it's such an interesting question. And we use the word decide, which is, I just think, so interesting. It's so anthropomorphic to think about a cell as a person making these decisions, like which type of breakfast cereal will I have in the morning. But it is true that to some extent cells often are faced with these sorts of binary, what we call cell fate decisions, where they're sort of faced with this question of, shall I do this route or go down this route? And what we often mean is that cells are turning on genes. So they might be turning on a gene that is related to neurons, for example, that might lead it down a more neural route, or they might be turning on a gene that's will guide it towards a more mesodermal route, which is muscle and some of the other tissues. So, yeah, I mean, these cell fate decisions are key to developmental biology, because that's how you go from the initial fertilized egg cell, a single cell, to something that has all of the cell types of the body that have to perform different functions. And in order to get that diversity, they follow these sequential fate decisions and then become all the different cell types that you need. But they have to do that in a way that is relative to each other. You know, they can't just make the decision in a kind of vacuum by themselves. They have to kind of look at their neighbours. What's my neighbour doing? Where am I in an embryo? Am I on the outside or the inside? And then they have to kind of integrate all of those factors when they're making their decisions as well, turning on the different genes. So it's an incredibly complicated process, and one that we're starting to understand, I think. But yeah, it's the key point of developmental biology. How are those fate decisions happening?
And from what you say, it sounds like that is something that, in a sense, sort of happens on the fly. It's not as though there's some predetermined instruction that's going to say this cell, eventually is going to turn into such and such. It's the cells are taking in contextual information from their surroundings, and I understand. Sometimes they kind of almost have to change their minds, or maybe literally, if we talk about it that way, have to change their minds. There's also movement involved, isn't there? That cells, they don't just decide where they're going to be. Some cells actually have to migrate to get to, if you like, where they need to be. It's really hard. I mean, it is possible really to talk about these things without giving it some sense. And I'm going to come back to this at the end of purpose or goals or something.
Yeah.
That process is it's, as you say, we don't fully understand it. But what really interests me is how seems to be this constant interplay between different scales of the system. At the scale of the cell, it's taking in, as you say, information from its neighbouring cells. Somehow that information is feeding back to the genes to turn some on and turn some off, and that in turn makes the cell do something new, and it's a constant building on that process. Seems to me that's what makes it so hard to talk about, because of that interplay. The idea that it's a plan that's there and it's just being read out is easy to articulate. The idea that you've got this conversation not just between different entities, between different genes at one level, between different cells at another, but between those levels is really hard to understand. I wonder whether it might help to talk about the very early stages of that process. So we start off with cells that kind of in the very early embryo that look identical. What do they do first of all to become non-identical? Is that something to do with, does the embryo still look uniform, but the cells kind of know they're different, or does the embryo shape change happen and then that determines what the cells do? How does that very early process start?
Well, so it's different in different species, but if we think about mammalian development, which I'm a marine developmental biologist, so I have a bias, that's my kind of sphere of interest. But then, so if we think about the fertilized egg cell, it's a single cell, it's been fertilized by sperm, and it will start to divide. So it will divide once, twice, a couple of times, and form smaller and smaller cells. So it divides without growing. And then you end up with this ball of a few cells, 30, 60 sorts of cells. And one of the first fate decisions is the cells on the outside versus the inside. So the cells on the outside are obviously have fewer connections between them. The cells on the inside have got lots of connections. They're surrounded by neighbours, and cells on the outside have fewer. And then there's also kind of polarity differences. So the way that the architecture of the cell membrane is different on the outside versus the inside. And so that's the very first fate decision that happens in the embryo. Are you an outside cell or are you an inside cell? And the outside cells will go on to form some of the structures that are important for implantation. Obviously, the mammalian embryo has to implant. So one of the first things the embryo has to do is find the uterine wall and kind of establish itself in there. And so the first cells set up all of those what are called extra-embryonic tissues, the tissues that support the embryo, even before they start to actually form their own sort of tissues of the organism, the brain and the skin and the muscle. The first thing the embryo does is form that extra-embryonic tissue. And so, yeah, it's a blob of cells with this outside cells and then inside cells. The second cell fate decision is once you start to form a cavity in the centre, so it looks a bit like a bubble at that point. You've got this kind of outer layer of cells and then this inner cluster that is going to become the actual embryo proper. And it's not really until beyond that point that you start to get anything like a form emerging. So you start to actually tell which side is your head and which side is the kind of tail end of the embryo. And then all sorts of interesting kind of rearrangements happen. So yeah, this cell fate decision, so the cell identity is happening at the same time as you're starting to get the shape changes, the morphology changes. You're also getting huge cell proliferation happening during this time. So it's, you know, if I was thinking about how to build an arm as an engineer, I'd probably say, well, I need a kind of structure first, right? I'll put the bones in place and then I'll put the muscle on top and then I'll do the outside stuff, the skin. But of course, the embryo doesn't start off with a big, fully formed arm. It starts off with something tiny that has to grow. So you're kind of coupling all of these processes together, the cell identity, the cell proliferation, and the kind of morphology, the shape of the structure at the same time. And it's an incredibly complex process. That's why we're still kind of unpicking it and trying to understand it.
And can you say something about the, let's put it this way, language in which the cells are communicating? As I understand it, there's at least three aspects of that language, that they're in contact with one another. They can exchange chemical signals. They can, in general, exchange electrical signals. And this is the one that kind of blows my mind when I think about it, because it seems so crude, and yet it seems so central. Mechanically, they feel each other. And if you tug on them, or if they stick together or are less sticky, that too affects
Absolutely.
What they will become, and it also affects how they might move around.
Yeah,
so is that right? Is that what are the languages?
Yeah, exactly. So the mechanical could be stretching, it could be compressing, it could be the actual extracellular sort of substances that are kind of between the cells. They can kind of feel what components are there. And yeah, I mean, it's something that I think the last sort of decade of research has really brought to the forefront. Like how are we understanding mechanical input to cell fate decisions? Because it's quite easy to say chemically, these are the signals that are around the cell, but actually unpicking what that mechanical input is much harder and more interesting, I think. Like you say, it's amazing that they are sensing what's happening in a physical way as well.
Well, I mean, I guess that also gives a feedback between form and cell type, doesn't it? That actually, if you have that mechanical sensitivity, then once you have tissues that bend or fold, that itself becomes a signal.
Yeah, exactly. And so your neighbour might be forming a flat sheet or an epithelium, as we call it, this kind of tightly packed sheet of cells. And then if you can feel that's happening, then maybe that affects your fate decision, or maybe that changes your access to different chemical signals. So we really have to understand not just at an individual cell level how the fate decision is happening, but how its environment as a whole is kind of changing at the same time. And that's obviously it's challenging, but I think that's the beauty of it, is the whole embryo is important to understanding an individual cell. And like you say, we have to work between levels. We can't just say, well, this is how cells build embryos. We also have to understand how the context of the embryo is important for the cells as well.
Yeah. Now, this is, I know this is a simplistic way of putting it, but it used to be thought for a long time that this was an entirely one-way process, that cells would sort of gradually, sequentially, work their way towards a particular cell fate, a mature tissue, like skin or bone or whatever. We now know, we've known for over a decade, that that's not the case. Actually, cells can be reprogrammed. We can take this process back all the way even to the early embryo. should you tell us a little bit about that and what difference that discovery made to the way we think about it?
Yeah, I mean, it was huge, right? I think before this point, I think people had assumed that as cells go through development, as development proceeds, that maybe cells were becoming sort of so specialized that they were then kind of trapped in that sort of specialization, right? And then as we started to think about genes, perhaps the cells were kind of losing some of this genetic information as they were making decisions, right? If I become a neuron, I only have neuronal genes, perhaps. But
so they actually thought they might be
chucking them out. Yeah, exactly. I don't need this anymore. But it became clear with John Gurdon's experiments that actually none of the genetic information is lost during development. So you can take a kind of later stage cell, and if you take out the nucleus, so take out the DNA, the genetic information, and put it back into an early cell, then you can actually start to kind of recapitulate some of that process. So nothing is lost in the process of development. The cells still have the same DNA, the same genetics, which actually I think is one of the most interesting findings, right? That all of the cells of your body, essentially, barring mutations, have the same genetic information, and yet they're so different. And so it's not that the actual DNA is lost or the genetics is different. It's just that the way that they're expressed, the kind of activity is different. And so then that, of course, opened up, well, if it's possible to take a later stage cell and take that genetic information and somehow reprogram that back to an earlier stage, then is there a way we could kind of artificially do that? And so, of course, Yamanaka came up with these factors that are enough to be able to take any kind of cell that is kind of somatic cell, so any cell of the body. So you could take a skin cell, you could take a hair cell, you could take a blood cell, and by applying these 4 factors, we can reprogram them back to an earlier stage. In fact, it's to a stage that is effectively embryo-like. So it's taking a cell right the way back up the decision tree. If we think about fate decisions as this kind of tree, right the way back to kind of embryo stage. And of course, that's incredibly powerful, because if you can take a blood cell, get it all the way back to the start of development, and then push it down a different route, so push a blood cell back to a neuron, for example, that opens up a huge amount technologically, but also just biologically. How do we understand our fate decisions if we can have that power of kind of going backwards and forwards at will? I mean, it's incredibly powerful.
And as I understand it, surprisingly easy. And I don't mean to underestimate the lab skills that you need to be able to do it. Yeah. But Yamanaka, so this is 2005, 2006, wasn't it? That he in the end had only to use to sort of give back to cells 4 genes that were active in the embryonic stem cell stage, which I think was a surprise to him, even, that you needed so few signals, genetic signals, to reset everything.
Well, yeah, I mean, you could imagine that it's an incredibly complex process going from something that's made all of its decisions right the way back to the beginning. I mean, it could have been thousands of genes that you needed to kind of kick back into gear, but it turns out it's just these 4. And I think that proves to us the kind of network structure of genes. So of course, a lot of these genes are transcription factors. So they're genes that encode for proteins that then regulate gene expression of other genes. And so just by adding these 4, they're kind of potent enough, if you like, that they target enough of the genome to be able to turn on, kickstart all of the other genes that you would need. So it's not just that the 4 are the only ones that are kind of differently expressed, but that they're enough to kind of kickstart the rest of the gene expression signature that you need for this early embryonic state. So, I mean, yeah, it was surprising, I think, that the genome is set up in such a way that it has this kind of plasticity, that just that 4 is enough to kind of hierarchically start that cascade off and turn a cell back to an earlier state.
Right. In parallel with that work, and partly as a result of it, I guess over the past 1 or 2 decades, people have been looking at how stem cells, whether they're reprogrammed cells or actually just normal embryonic stem cells, how they develop in culture, in a petri dish, into larger tissues. And cell culture, I mean, it's a century old now, but what seems to happen in these cases, if you grow them in the right way, it's as though cells, if you guide them towards a particular cell type, it's as though they somehow have the innate knowledge of, oh, I need to grow a gut or a brain, and they start to develop along those lines. So this is the science of organoids.
Yeah.
Can you tell us a little bit about that? And in particular, I'm really interested in how we can talk about that kind of what seems to be that innate knowledge, not just of what cell type to become, but of the morphology of the tissue that is dictated by that choice.
Yeah, I mean, that's a lot to unpack. I think finding these IPS cells, as they're called, these induced pluripotent stem cells and the embryonic stem cells, opened up, as you say, this field of kind of cell culture and us understanding better which signals were required in order to make cell fate decisions. So I think that's a kind of key point in our understanding, really. As soon as we could grow these cells and culture these cells, we could learn what signals that they needed. And so our first kind of approach, I guess, as a field was to say, okay, well, if I add this chemical signal and then I add this chemical signal and then I add this one, can I make a neuron or can I make a muscle cell, for example? And that, to some extent, works, right? That's an incredibly powerful way of forming a different cell type that we want. But of course, that's a particular cell type by itself. And we've already discussed, right, how important it is that you have the context. Development develops as a contextual information. And of course, in an embryo, these signals are not just kind of applied at kind of 24-hour intervals like we might do in a laboratory, right, where we've got these kind of set up conditions. In an embryo, the signals are coming from the neighbouring cells or from the cells themselves as they're making these decisions. And so I guess a lot of the logic was to say, well, rather than us imposing these chemical signals kind of exogenously, because that's how we do it in the lab, what happens if we let these cells grow by themselves and interact with each other by themselves as they would do in an embryo, right? There's nothing kind of weird or unnatural about it. It is just what cells do in an embryo. They interact, and that's how they make cell types. So that seems logical that would also work in culture. And of course, when you set them up in such a way that they can interact with one another, so often that means in aggregate, so when they're talking to one another rather than separate, and in suspension, so when they're floating, free-floating as aggregates, then they often will form what are called organoids, as you say. So if you take an early sort of intestinal sets of cells and put them together, they might make intestinal organoids, or cerebellar organoids, so brain organoids from early neurons. So yeah, I mean, it's an incredible discovery, really, this idea of organoids, because it relies on this idea of self-organization, that rather than us being the ones to impose signals or to impose sort of this order or direction, if you like, the cells themselves are coming up with it. And so I guess the kind of old analogies that we have for cells, things like them as the bricks of a house or as the kind of building blocks, it kind of doesn't get across that idea of, I mean, as you described in your book, agency, or this idea of kind of working together in a kind of dynamic way, that of course the cells are not just changing over time by themselves, but interacting with each other. As they make a decision, then they might be signaling to their neighbours. That might influence their neighbour's cell fate decision. That might influence their other neighbour. All of these things are happening at once, all together. And so I think, yeah, it's been amazing, the organoid field, how it's taken off, and it's really shown us self-organization. How is it that these cells are forming structures, forming patterns, and doing that in the way that teaches us about how the embryo does it? Right. So yeah, I mean, how do we talk about this? I'm not certain that we've got the perfect terminology. Like I say, I think self-organization is key. And this idea of sort of feedback, if you like, between cells and information processing, that it has to be cells working together as an ensemble to make that pattern.
Right. And I guess the things that they make, like you say, if you're doing this with gut cells, then they might make a sort of hollow tube-like structure. With the brain, you start to with neurons, you see something brain-like, but it's not exactly as it is in the developing embryo, because the other tissues aren't around there, and we tend to talk about it as well, as though they're trying to make a brain, but they don't really know how, and they make an imperfect one. But of course, there's nothing imperfect, there's nothing wrong with the structure they're making. It's what those cells will make in that circumstance.
Yeah.
And it gives us a kind of sense, I suppose, of there being more plasticity in form than we see particular kinds of form in the developing embryo, but that's not the only, that's not the limit of the forms that the cells can make.
Well, exactly. And the embryo is such a kind of constrained scenario, right? It's got to form something at the end that is effectively viable and fertile and the subject of evolution. And all of these things are kind of constraints on what the embryo can do. And of course, once you start to break that apart or take it apart, then you lose some of that constraint. And maybe that teaches us more about the actual logic that the cells are using. So I really think that actually, with a lot of these experiments, you learn more from how they're different to embryos than how they're similar. I often talk to people who say, well, look, I've got this model, it's exactly the same as X, Y, Z that happens in the embryo. And I think, well, that's a shame. You want it to be a bit different, because then we learn, oh, well, actually, this thing is very similar to an intestine, but actually it doesn't have this particular cell type. Okay, so what are we missing in our in vitro culture that is different to how the embryo does it? And can we then play with that, add that back, and then figure out, oh, okay, well then in that case, what the embryo is using is X, Y, Z in order to make Y. So yeah, I think there's a huge amount to be explored in that hidden space. I mean, people talk about morphous space, right? This multi-dimensional kind of space in which form might be found, right? So there's the forms that we see that happen in real embryos, but there are also these kind of hidden forms that might exist that could potentially be unlocked with some of these kind of unconstrained embryo cultures, for example, or organoid cultures. So yeah, there's a lot to be explored and a lot to be understood.
Right. Well, we've started to touch on this already because as well as organoids that are growing to be somewhat like actual organs, we can do this with the whole thing, and this is what you're doing with the whole lot. We can do it with embryos, because if you have something that is pluripotent, that is able to grow into any tissue type under the right circumstances, and maybe you can say a bit about that, they should be able to grow into something that looks like an actual embryo. So can you explain how that's done and what it's possible, what it has been possible to grow that way so far?
Yeah, so of course this is the field of kind of embryo models or stem cell based embryo models, and yeah, like organoids, it relies on the same principle, right? That cells interact with one another to self-organize into structures that we see in biology. So organoids typically are one particular organ, as the name would suggest, and then these embryo models, or embryoids as some people call them, often have multiple lineages of cells. Sometimes they are just the kind of embryonic portions of the actual embryo proper. Sometimes they have some of that extra embryonic tissue that we were talking about. So there are different ways to make different embryo models. There's a huge plethora of different embryo models. But yeah, all of them are kind of playing with this idea of wanting to understand how cells make embryos. And so using these principles of self-organization in vitro in the lab in order to understand better how that process might happen. So some of them, for example, as I say, might have these kind of extra embryonic tissues that make them look very similar to kind of blastocyst stage embryos. So this is the stage, as I say, when the embryo looks a bit like a bubble. It's just about to kind of implant into the uterus. You've got these outside cells and then a cavity, and inside there's the inner cell mass, the inside cells. Other models might be kind of replicating a slightly later stage of development when the cells are starting to form more kind of fate decisions like mesoderm or neural tissue. So there are different stages that we can replicate. There are different kind of complexity of the models, if you like. But all of them are trying to get at this idea of, well, how the cells build embryos, and can we learn about that logic with in vitro culture?
And as I understand it,